Today we're very pleased to have with us Dr. Ian White, has been an expert in regenerative medicine, both in academics and in industry, and he's been involved in many exciting aspects of the field. And today he's going to be shedding light on some of the myths and ideals surrounding both autologous and allogeneic products and their use in regenerative medicine, as well as some interesting concepts around coronavirus, COVID, and inflammatory diseases. So, Dr. White, thank you very much for being here with us today. We're always happy to hear from you.
Well, thanks very much, Joe, for that introduction. It's a pleasure for me to be here today. It's a very novel and interesting way to reach everybody. So I'm very happy to be here and talk a little bit today about perinatal tissues. I wanted to do something a little bit different this time, and I wanted to address some of the fact out there versus some of the fiction that is circulating relating to perinatal tissues and regenerative medicine as the whole. So hopefully there's going to be informative. I'm looking forward to any questions that may arise down the road. Let's jump straight in with an introduction to myself for those who don't know me. My name is Dr. Ian White. I am a regenerative medicine scientist. I received my PhD from Cornell University in the Ansari Stem Cell Institute, the Division of Regenerative Medicine. And since then, I've been working in academia and now commercially in the space of regenerative medicine, primarily focused on developing perinatal tissues for clinical research and clinical trials. So the name of our company is called Neobiosis, and we are an FDA-registered CGMP-compliant biotechnology company. And we have an R&D laboratory located within the University of Florida Innovate Biotech Institute. So we're very unique in that sense that we have not only are we manufacturers, but we also have our own research and development arm in-house. So our R&D lab is based in Alachua, Florida, and we have our manufacturing clean rooms located within the UF Innovate Hub in Gainesville. So our work is grounded in science. That's our focus, is looking at regenerative medicine with a scientific eye, with the goal of commercializing the next generation of cellular and acellular regenerative medicines for research and clinical trials in a global market. So I'm going to do this a little bit different, a bit of a departure from what I'm used to. I'm going to try to predict some questions out there, address some misnomers with some scientific fact. And I've included a disclaimer here to address that. So, of course, all facts are subject to the scientific process. And as such, some facts may evolve over time as our understanding of regenerative medicine expands. So everything within this presentation is, as far as I'm aware, scientifically accurate as of today, but those facts may evolve over time as we learn more. So starting off with the first misnomer, in this industry, there's an overuse and a misuse of the term stem cells. So often in this field, stem cells are thought to be synonymous with regenerative medicine, and that's just not the case. So we really need, as an industry, and I've said this many times before, but we really need to start using terminology correctly if we're going to push the field in the right direction. So stem cells and regenerative medicine are not synonymous. They are not the same thing. While some products do contain stem cells, there's actually no scientific data in support of the idea that undifferentiated transplanted stem cells contribute to regenerative medicine. So as we're going to learn as we move through this presentation, stem cells do not contribute directly to a tissue that is diseased or injured. What they do is they contribute to the signaling that promotes the endogenous repair. So the body's own ability to repair is stimulated by the cells that are transplanted, and those cells are not stem cells. One of the issues that a lot of people in the field end up getting into is something like what happened with this company here, where they were advertising the use of stem cells. They were not appropriately trained, and they ended up hurting blinding three women. We have to be very careful in this industry to not only perform the interventions correctly, but also to not misinform patients and not misinform the industry as to what we're doing. We are not in the business of selling stem cells. We are in the business of understanding regenerative medicine. And again, they are not the same thing. On a related note, MSCs are not stem cells. Many reps will give you the idea that the stem cell therapy that patients are asking for can be found in a product that is replete with MSCs. This is not the case. MSCs, while very well studied, are not stem cells. They're very well studied in the field of regenerative medicine, but they are not stem cells. The name for them is still under debate, but the names that we typically use to describe MSCs these days are mesenchymal signaling cells, mesenchymal stromal cells, or medicinal signaling cells. And you don't have to take my word for it. You can pull up this article from Arnold Kaplan, who is considered the grandfather or the father of MSCs. He discovered, studied, and named MSCs. And back in 2017, he agreed with the rest of us where we argued that MSCs are not canonical stem cells. And in this paper here, he makes a case for changing the name of MSCs from mesenchymal stem cells to medicinal signaling cells. And here he says that it's to more accurately reflect the fact that these cells home in on sites of injury or disease and secrete bioactive factors that are immunomodulatory and trophic, meaning that these cells can make therapeutic drugs that are medicinal. The idea there is that the MSCs secrete growth factors, etc., that contribute to regeneration by stimulating the endogenous cells. So these are not stem cells that differentiate into damaged tissue. These are medicinal signaling cells that release signals to instruct the endogenous tissue-specific stem cells, perhaps, or tissue-specific mature cells to mediate repair. So often there's a lot of confusion with the names that are floating around. For example, people will often use terms interchangeably like stem cells, exosomes, SVF, which is stromal vascular fraction, bone marrow, umbilical cord, cytotherapy and cell therapy, regenerative medicine. These are not the same thing. They are all very specific and all very different. For example, exosomes are not cells. Many times I hear people ask about exosome cells. They're fundamentally misunderstanding the concept of exosomes, which are a derivative of cells, often from stem cells, often from MSCs, often from other cells that are involved in the regenerative process. And they're used as facilitators of signaling. So messages are packaged within exosomes that are then released and liberated from cells, and they express markers on their surface, which they use to find and address, for example, diseased or damaged cells within the host, and they're able to adhere and release their cargo, their messages to those cells. So they're not cells themselves, they are products of cells. SBF doesn't contain stem cells. You'll hear a lot that people believe that the SBF products are replete with stem cells. What they're referring to typically are MSCs, but as we've just discussed, MSCs are not stem cells. Bone marrow contains hematopoietic stem cells, which are canonical stem cells, and MSCs. But what we understand about science is that it's unlikely that those hematopoietic stem cells that are undifferentiated actually contribute to any of the regenerative therapy. The idea there is that they're just not mature enough. They don't have the opportunity to mature. They don't receive the correct signals in order to be able to release regenerative signals that would mediate repair. It can take a long time to differentiate those, and we'll go into that in a second as well. So regenerative medicine generally is cellular or acellular therapy. So it is not stem cell therapy. It is acellular or cellular therapy. Various different cells can contribute to regenerative medicine. It is not a stem cell. This is not a stem cell industry. So another misnomer out there is that autologous cells, so the patient's own cells, are the best option in regenerative medicine or regenerative therapy. Now, while SBF and bone marrow do have demonstrable regenerative potential, they have been demonstrated in many clinical trials to have strong and potent effects when it comes to repairing tissue or modulating the immune system, reducing inflammation. That's typically only when the bone marrow or the SVF is very young. And for most patients, they are typically older. And what we're noticing when older patients' material is used, so bone marrow and SVF, is that these cells typically don't contribute to the regenerative process, but what they do is they provide an irritation. So much like prolotherapy, these cells that are being injected from older patients, they've lost the ability to regenerate, but what they can do is act as an irritant, again, like prolotherapy, and that's where we see the benefits. So here's an example of why we focus on perinatal tissues. So in the newborn, the fitness or the ability of those cells to contribute to regenerative medicine is very, very high. And that ability drops precipitously after birth. And if we're looking at the 50 and 60-year-old patients, those bone marrow cells or those adipose cells have very little regenerative capacity remaining in them. And again, what they do contribute then is just an irritation. So it's a very expensive, very painful way to administer an irritant, but that's primarily the mode of action of those cells in the aged population. So we prefer to stick with the newborn products that are replete with regenerative medicine. And as we can see here, this is some data that we pulled from our website. So if you go to www.neobiosis.com, this is a data set on our Wharton's Jelly cellular product. And as you can see, there are over 200 cytokines and growth factors and soluble receptors that are present in this product. And I've sort of highlighted just a few here to give you an idea of what's present. But if you want to see the full list, you're able to go to the website and check that out. So this very young perinatal tissue is absolutely packed full of growth factors and other proteins that can modulate and mediate an immune response or modulate a reduction in inflammation. So another idea that I hear circulating around is that it's only the MSCs that have regenerative potential. And this clearly is not the case. While MSCs are definitely the most studied cell when we're talking about the context of regenerative medicine, they are certainly not the only cell that participates in regeneration or even the most potent. So what we understand about regenerative medicine is that it's more like an orchestra. A single cell is not responsible for mediating all tissue repair and all immune modulation. There are many cells throughout the body that contribute to a regenerative response to injury or a inflammatory response, not just MSCs. So one thing I hear very often from companies that are manufacturing products from umbilical cord is that they claim to have millions and millions of MSCs per inch of umbilical cord, and this just simply is not the case. Now, testing for MSCs is very, very difficult, and often people, non-scientists, will make errors in understanding what they're looking at when they're looking at cells from various tissues. So if you take an appropriately trained professional flow technician, you can see quite clearly using the appropriate techniques that there are about 400,000 MSCs typically per inch of umbilical cord, and that's about 10 to 15% of the total viable cellular component of an umbilical cord. And what that ends up translating to typically is about 40,000 MSCs per 1 cc vial of a Wharton's Jelly cellular product. Now here's some data. We trust third-party independent labs with our data because we want to make sure that everything is done external. We don't do anything in-house that could contribute to bias, so we do everything external. And this particular data set is generated by the University of Florida at their flow cytometry core. And using a very specific panel of markers where we work very closely with Beckman Coulter in order to design this panel, we were able to find that we have about 12% in this particular cord, 12% MSCs, and the remainder are EPCs and endothelial cells and some leukocytes, as you can see here. So one other thing you might hear is that umbilical cord tissue only contains MSCs. And as that data set just demonstrated, that's not the case. Umbilical cord cellular products will typically contain a lot of different cells, many millions of cells, but only perhaps 10 to 15% are MSCs. The rest are demonstrated here. So this is from our website again, and you can see that we have a lot of different cells when we're processing an entire umbilical cord. We have human umbilical vein endothelial cells, we have epithelial cells, we have human umbilical artery smooth muscle, and we have MSCs, and we certainly do have some hematopoietic cells in there as well. So we have two different products at Neobiosis that we design. One is a Wharton's jelly cellular product that's like just described, and the other is a Wharton's jelly acellular product. So it's just the Wharton's jelly matrix. And in that product, what you might find is a lot of extracellular matrix, certainly a lot of EVs, so extracellular vesicles, and it's replete with all the growth factors and cytokines. Again, the same or similar panel of growth factors and cytokines are present in both the Wharton's jelly matrix and the Wharton's jelly cellular. They both also contain a high level of hyaluronic acid, which is critical if we're going to be looking at clinical trials for knees and degenerative joints. But the cellular product is unique and different from the matrix in that it contains all of those cells that I just described as well. So these are viable, live cells that will interact with the environment and theoretically and hopefully respond to that inflamed or damaged environment by producing a unique set of proteins and cytokines. So one thing I hear a lot is that there are no live stem cells in commercial regenerative products, and that only bone marrow shows live cells when you look at the CFU, so colony-forming units. There are several people around that publish research out of their makeshift labs where they try to demonstrate that only with their bone marrow technique can they get live stem cells. Well, what's apparent, especially when you've spent so long at the bench working with these kinds of cells, is that for the most part, the cells they're looking at are fibroblasts. So they're not stem cells. They're quite often fibroblasts. And if they're not fibroblasts, they may be MSCs. But again, we've already discussed that MSCs are not stem cells. So the stem cells that you might find in bone marrow or umbilical cord, so perinatal commercial regenerative products, these hematopoietic stem cells can be cultured. But they're very, very hard to culture, and it takes a lot of expertise and knowledge in order to do so. You can't just buy a media online and throw the cells into a Petri dish, into a 37-degree incubator, and expect to see those stem cells growing out. It's just not done. And in fact, it's so difficult that it was the focus of my PhD thesis. I spent a lot of time focused on designing ways to culture expand hematopoietic stem cells, and I was successful. I'm going to show you that in a second, but I will say that stem cells, if they're used in regenerative medicine, must be activated and differentiated first, and this can often take days or weeks in culture. So any hematopoietic stem cells or any other stem cells that a rep might be claiming that are present will likely not contribute at all to the regenerative process. And if you're using stem cells in a clinical trial, those stem cells are typically differentiated into a tissue type. So they're matured to a very specific tissue type over weeks in culture before getting that definitive cell type that's then used in the clinical trials. So stem cells, yes, are used in regenerative medicine clinical trials, but they're differentiated first. So here's an example of culture expanded hematopoietic stem cells. So these are long-term hematopoietic stem cells. And what you can see by day 7 is that there are very few cells in that top left panel. And then if we look at by day 28, we've got a lot more cells. And the green is a GFP notch. So notch is just an indicator of stemness. So what we're getting is not only an expansion of the cells, but also a proportion of stem cells is increasing relative to the non-stem cells. And you can maybe see on the bottom panels there that these round hematopoietic stem cells are cultured on a feeder layer of endothelial cells. And what's important is that this has to be done without serum, because serum will cause the cells to differentiate and then they won't grow. Again, many other groups aren't aware of this, and when they're trying to demonstrate that there are no stem cells present in various products, they pour the product out into a typical canonical culture media that contains serum, because most cells require serum, and then the cells mature, differentiate, and don't divide anymore, and then they claim that there are no dividing colony-forming unit stem cells present. So there is a technique involved. It is challenging, but this does demonstrate that there are many hematopoietic stem cells in not only bone marrow, but also cord, blood, and cord tissue. So I hear this often also, that there are no live stem cells in amniotic fluid products. There are certain individuals that talk about this quite often publicly, and they've hired prominent scientists to demonstrate also that there are no live stem cells in amniotic fluid. And it's sort of used as a gotcha by many sales reps that are trying to sell other products that are not perinatal products. But it's quite funny because, of course, there are no stem cells in amniotic fluid. That's sort of the point. Amniotic fluid products are designed and derived to be acellular. It is a liquid that contains extracellular vesicles, and it is completely devoid of cells. The cells are any cells that might be present in raw amniotic fluid are removed, and you're just left with amniotic fluid, EVs, and other soluble proteins. So here's an example then of what amniotic fluid is. So amniotic fluid is derived from the amniotic sac at the time of birth of a natural, healthy cesarean section under appropriate protocols by the following FDA and CDC guidelines and with consent from the mother. And what we're able to derive is a large number of extracellular vesicles that is suspended in a medium of growth factors, cytokines, lipids, complex sugars, peptides, and urea, which are all very good for tissue growth. And also they're isolated during a period of intense tissue growth and immunomodulation. So these are trained to not only grow tissue, but also modulate the immune system. And amniotic fluid, as we know, is very antimicrobial. So this is also very good for sterility purposes. We have a 100% sterility record at Neobiosis, but also it's very good if you want to use it for an antimicrobial purpose in one of your clinical trials. And what's very exciting is that unlike perhaps culture expanded exosomes, which are derived from a single cell source, these EVs are derived from a heterogeneous, diverse cellular population. So the secretome is very diverse, and we get a very broad spectrum response, which is more like an orchestra playing. When you want to regenerate tissue, you want the full complement present. You don't want to be restricted to a single cell type. You want the full orchestra present, and that's what we find in amniotic fluid EVs, or what we call cytosomes. This is a very common misconception, or statement, I should say, that amniotic fluid is no more than baby pee. Now, while it's true that actually the majority of the liquid in amniotic fluid is derived from urine, from the fetus, that's actually a good thing. As I mentioned, there's a lot of good stuff in that liquid, growth factors and everything that's required for a developing fetus. And also it's replete with the EVs, the cytosomes, that contains over 300 bioactive proteins, cytokines, and soluble receptors. And we have a full list on our website. If you want to visit neobiosis.com, you can see a full list of bioactive proteins that are present in amniotic fluid once it's purified. And you don't have to take my word for it. You can search the literature. Here's just a snippet of a few articles talking about the regenerative potential of amniotic fluid, and that amniotic fluid isn't really just considered fetal urine anymore. It's really being recognized as the frontier tool in regenerative medicine, and that's why we like to focus on it here at Neobiosis. To generate cytosomes, we have a proprietary process where we collect amniotic fluid from an AATB accredited cord blood bank, and we receive that product within 24 hours, and we process it in our ISO 7 CMG compliant clean room in Gainesville. And after a series of filtration steps, we end up going through a double sterile filtration at the end, and we're left with a very pure product of EVs that's around 50 to 150 nanometers in size. And that's very important, because then we know we've got the EVs that are the signaling tools used by cells to communicate to one another. Those small EVs between 50 and 100 nanometers are the ones that contain all of the messages, the RNA and the protein that we want to deliver to the regenerative site. And the way that it works is very fascinating, actually. So the reason that exosomes or extracellular vesicles are so small is actually a physical feature. It's actually required in order for them to function correctly. So if they were any larger, they wouldn't be able to deliver their cargo. So there's a phenomenon called Laplace law, where a very small vesicle, a membrane-bound vesicle that's floating by a cell and has adhesion molecules on its surface, recognizes the target cell through a receptor ligation, and then it's able to merge with the membrane. And the only reason it's able to liberate its cargo within the cell is because of this phenomenon of Laplace law, where the internal pressure is inversely proportional to the radius of the sphere, which means that the pressure inside the EV is gigantic compared to the pressure inside the cell. And so what that means is when the membranes merge, the cargo within the exosome is shuttled into the cell, pushed into the cell because of the immense pressure inside that small vesicle relative to that of the cell. So once the cargo, the RNA or the protein is delivered to the cell, the cell is able to utilize it. So whether it's a mature protein, the cell is able to utilize it immediately. If it's an RNA, that RNA is able to find a host ribosome and become translated into a mature protein. So what's very exciting is that those EVs from perinatal source contain all of the young signals that are required to pull back the regenerative ability of the body to that fetal stage. So as we age, we lose the ability to regenerate tissue. But if we take signals from perinatal sources, all of those RNAs and proteins that are present in the fetus that contribute to the ability of the fetus to repair and regenerate and to modulate the immune system are present in these EVs. And those RNAs can become mature proteins within the older cells. And so if we're talking about collagens, these products could be theoretically used for cosmetic purposes, because one of the reasons why our skin starts to sag is because we no longer have the full complement of collagens that we had when we were younger. But these EVs are able to deliver those collagens. Our cells are then able to translate the RNA into functional protein, and we're able to make fetal and young collagens again. The same with other proteins. Same goes for other cargo, whether it's VEGF or FGF, which are fundamental in angiogenesis, which is critical for tissue repair, or IL-1 receptor alpha, which is important in immune modulation, and stem cell factor, which is important in recruiting endogenous stem cells to sites of injury. So all of these different RNAs can be present within these exosomes or these extracellular vesicles, and they can be delivered to target cells because of this phenomenon of Laplace law. Functionally, we have some data here that demonstrates that our cytosomes not only contain a lot of growth factors and other signaling peptides, but that those growth factors and signaling peptides are functional. They actually do something. They work when they're introduced to tissues. And so this in vitro experiment was performed by ZenBio, so an independent third lab. And what we're looking at here is what's called a wound healing assay, where we take a layer of epithelial cells that are culture expanded, and there's a small drop of gel that's placed in the middle. And when the cells become confluent, so when the entire plate is full, that gel is removed. And if we don't add any growth factors, so that panel here, which has no growth factor control, if we don't add any growth factors, nothing happens with those cells. They just sit there and they start senescing, and they act like they're old. But if we add growth factors that we know can activate epithelial cells and can mediate repair, so in the very right-hand side, growth factor control here, what we can see is that those epithelial cells activate and they migrate into the wound and heal the wound. Much like what we see with cytosomes on the left, if we go 18 hours after administering cytosomes to those cells, the extracellular vesicles are able to bind to the cells, deliver their cargo to the cells, and those cells are able to activate and migrate into the wound and repair the wound. So this is a clear indication that the cells are receiving all of the signals that they require and the building blocks in order to activate, divide, and migrate in response to just the presence of the amniotic fluid cytosomes. Here's some in vivo data. So again, we like to partner with external labs so that there's no bias. Here we're working with Harvard University, and what we've done is a typical experiment for regenerative purposes, which is a paw incision. The mouse receives a small cut in the paw and either receives cytosomes, IV, or saline. And what you can see over 10 days, there's 4 different mice here. Over 10 days, the mice that receive cytosomes appear to heal much faster than those that didn't receive any cytosomes. So this is clear in vivo data that supports the in vitro data that the cytosomes present in the amniotic fluid are active in contributing to the regenerative process. Here's some more data from the same lab at Harvard, where we took Wharton's jelly matrix, or we took cytosomes in an arthritis induction model. So the animals were induced to develop arthritis experimentally, and then they were administered either Wharton's jelly matrix, cytosomes, or a control, which is just saline. And what you can see is that those control animals, although they did naturally repair and recover slightly, the recovery was much more robust when we introduced Wharton's jelly matrix to that joint, or cytosomes to the joint. So clearly both products and their growth factors and their EVs are contributing to not only experimental in vitro activation of cells, but also they're reducing the inflammation in an arthritic knee in an experimental model here. Yes, it's using mice, but it's a very good indication that we will have success when we attempt a clinical trial with humans. What's next for allogeneic perinatal products? Well, where we want to go is somewhere that bone marrow and stromal vascular fraction can't go. And at this time, it's COVID. So when we're talking about COVID and recovery from COVID, we can't use bone marrow, we can't use SVF from the patient because they're already sick. And so what we need to do is find an alternative method for reducing the inflammation in these patients and promoting tissue repair in the lungs. And so we were featured heavily on the news recently for our ongoing work on treating long hauler syndrome, which is a symptom of COVID after patients recover. Up to about 80% of survivors can end up with at least one and usually more symptoms that are ongoing. They appear to be long term, and it's unclear at this stage how long a lot of these symptoms may go on for. But many patients, again, up to 80%, even if the patient has had very mild symptoms during the chronic phase, the post-COVID long haul appears to affect many, many people in many, many different organs. People are experiencing hypertrophy of the heart. So there are people still dying of cardiovascular disease as a result of COVID, even though they had very mild symptoms of the virus itself. But patients are presenting with gastrointestinal issues and a lot of brain issues. So a lot of nerves become affected with the COVID virus after the vasculature becomes fenestrated and leaky. The immune cells can leave the blood vessels, migrate into the nerves, and cause all this organ-specific injury. And so we want to be able to treat that because it's going to be a major issue moving forward. There appears to be no age distinction associated with the outcome of post-COVID syndrome. Young people are affected as old people are affected. There is a distribution in males and females of 4-to-1. For some reason, females seem to be more prone to having more severe post-COVID syndrome symptoms than the males do. That's something we're trying to understand. But all these symptoms seem to resemble things that we've seen before with other viruses. So it's a post-viral syndrome is very common after viruses such as Ebola and even the flu, where patients can be sick for a while, but that usually resolves relatively quickly and is usually not fatal. There are some cases where these issues become chronic, especially in the case of CFS or ME. These patients can suffer for a very long time, and we don't want to be seeing that with COVID. We don't want to see a whole new generation of individuals, especially if it's up to 80% of people that are going to be exposed to this. We don't want to see that going on and affecting an entire generation. So there's no approved treatments currently, but we are in the process of developing a clinical trial to administer cytosomes for post-COVID syndrome. And some of the reasons why we think that that's going to be successful are that there's been a lot of research over the years of how amniotic fluid contributes to the development of the pulmonary tissue. So without amniotic fluid, in a lot of different models, what scientists have found is that the lungs just don't develop properly, and we get a lot of pulmonary hyperplasia in the absence of amniotic fluid. So it's clear from a development perspective that amniotic fluid and the EVs and the growth factors that are present in amniotic fluid are critical for the growth and health of lung tissue. And so for that reason, we're very optimistic about the potential outcomes of this clinical trial using amniotic fluid to treat long hauler syndrome and to treat those damaged and inflamed lungs. But there's some additional data that's coming out that's very exciting and again makes us very confident that our clinical trial will be successful. In the news recently, an Israeli group was claiming to have found the cure for COVID, and they say that their product ExoCD24 is very effective at treating COVID patients. And in fact, 29 out of 30 of their first test subjects that were very severe ended up being discharged from hospital after receiving these exosomes that were enriched for CD24. So that's their process. They take exosomes, they enrich those exosomes that are high in CD24, and they administer that as the treatment. And what's very interesting is that we already know that amniotic fluid is very high in exosomes that contain CD24. So, of course, we wanted to look to see what the status of CD24 was in our cytosomes. And again, a third-party independent lab demonstrated that, yes, our cytosomes are extremely high in CD24. And in fact, we have between 4,000 and 6,500 picograms per mil of CD24 in our cytosomes. So if the Israeli study is anything to go by, that EVs that express CD24 have a very potent efficacy when it comes to COVID, we have high confidence that our product will be successful in treating it. So we're very, very excited to move forward. Hopefully that was an informative overview of some of the misnomers in the perinatal regenerative medicine space. Hopefully I clarified a few questions that you might have as to what the differences between the products and what the products can and can't do. And with that, I'll close.
Ian, thank you very much for that great presentation. I'm always amazed at how you're able to accumulate the information from the industry and put it in a meaningful way that people can understand. And I think the products that you're developing and the ideals that you're embracing relative to how the machinery of regenerative medicine works are very important. Everything from cell signaling and exosomes to their abilities to fight inflammatory diseases is more relevant now than ever, certainly in light of what we've been through as a country in the world regarding the coronavirus, but also opportunities that these products bring to the industry and patients in need across a wide spectrum of clinical applications. So, as always, it's wonderful to hear from you.